But okay, great. Sorry for the delay. I'm sure now everyone's rolled in, but yeah, it's been a long but very fruitful and interesting day, and kind of humbled to have these faces on screen: Dr. Kumar, Dr. Hansen, Dr. Bank, yeah, everybody here. So I guess let's I guess roll right into it. I guess people can leave questions in the chat just as you could for all the other sessions. But I guess so. You've all kind of had your sessions, and there were a lot of questions that probably went over. Maybe I should start with for each of your sessions. Is there anything that you kind of wanted to kick it off? Anything each of you wanted to kind of express that you weren't able to? In the question sessions, maybe in order, Dr. Kumar, if you want to start with that.
No, I think probably obviously I couldn't get kind of whole time to express the full model or just kind of explain the whole model. But I guess I kind of like express the crux of the argument that we are trying to make, going from the sound model to the source model, and obviously it has lots of implications. It has lots of implication in terms of what effective therapies could be, for example, designed around that one. For example, why the mimicry works for some people, why it doesn't work for other people, and so I think in terms of obviously I'm not a clinician, but I think what could be if this kind of model works and it is applicable, so it can be possibly translated to some of the possible therapies also, actually. Yeah.
Gotcha. Okay. And actually, yeah, well, I was going to go to Dr. Hansen next. Dr. Brown reminded me that. So I think you had a question for the last session that I didn't get to ask the speaker. Is there something that you'd like to ask now as part of the panel?
Yeah, I totally forgot what my question was.
Oh,
Dr. Brad, if you remember my own question, that'd be great. I don't know. Come back to me.
Yes. You had asked, I thought it was a great question. You had asked Dr. Davidenko that if,
yeah.
I'm trying to remember it now, if the alternative video was played with the trigger sound, could it work it in the reverse? So in other words, do you remember the question now?
Yeah, I think I kind of remember touching on it at least a bit. Yeah, just wondering how it affected the next stimulus. And I think Dr. Davidenko gave a reply to that a bit. But obviously it could be possible that it makes it worse, but it's hard to tell for sure, I suppose.
Right, one of the many, I think, further studies that can come out of that.
Yeah.
That resolves. Well, speaking of further studies, I know I'm jumping around, but Chris had a question, and I think we're all probably wondering: a dream upcoming study. If kind of money was no object, what would each of you like to see studied next? And I'm sure the Misophonia Research Fund can make that happen. But Dr. Hansen, do you want to start with kind of like what your kind of your dream research study?
Was that to me? Sorry. Yes.
Yeah.
Happy to jump in here. One thing I've always wanted to do is do a more personalized approach to misophonia, because as we've all probably experienced as we collect data, the sounds we use might not be the sounds that bother our participants the most. If it is a condition where their mom or their dad bothers them the most, is doing a actual collection of personalized stimuli, and that could be in the form of carrying around a recording device or whatever it is, but have participants spend their day collecting their own stimuli and using their own sounds in an experiment compared to, for instance, another participant's sounds to see if it truly is person specific or if it's like in general you'll be triggered across the board chewing, that kind of thing. Something in that realm would be cool.
Gotcha. Yeah, and Dr. Guzick, your kind of dream study, and feel free to everybody jump in to kind of like counter or suggest.
Holy cow! Yeah, it's a great question. my mind immediately goes towards treatment development, which is a very expensive process, and it's often rushed because it is so lengthy and extensive. But if you had unlimited resources and you could really plan out a 10 or 15 or 20-year program of research, you could really get into really understanding, one, the core components of interventions that are effective, one, and two, why they're effective, and then three, making sure we're testing them in settings and with people who will really use them. And all three of those things, understanding what works, understanding why it works, and understanding how it can get out there, are like entire research endeavors that are extensive. And it's sometimes hard to know how to prioritize time, resources, etc. But if we could get all those things together, there's now been so much proposed, and we do have this rapidly developing understanding of the mechanisms. I think we're on strong enough ground based on misophonia specifically and related problems and what's been working for those, that we could start really getting into the details of what works, why, and how to get it out there. That'd be what I'd be thinking about.
Sarah, how about you?
Yeah, absolutely. I think all the points that everyone's made, I agree with. I think for me, coming from sort of a more neuroscience perspective, it would really be great to have the opportunity to be able to partner with some clinicians and kind of create some sort of study where we're thinking about these mechanistic approaches and how those might be applied in a clinical setting. And also, to Dr. Hansen's point about sort of the personalized approaches, I think it would be amazing just to be able to get sort of like a large enough in-person sample where we can really look at their subgroups within misophonia. That's something I've been very curious about, whether it be by type of trigger or by type of reaction or things like that. Those types of studies require so many participants, and I think it's really hard to achieve that. But if we were able to kind of work together and pull data and do some sort of study where we could get those large numbers, I think that would be amazing.
Gotcha. Question from Mary to come on a couple sessions together. Question for Dr. Kumar and Dr. Davidenko. Referring to the source model as opposed to the sound model, would the sound swapping of Dr. Davidenko's experiment align with the source model for explaining misophonia? And is there an emerging sound model that is also promising?
So is that a question for me, sorry?
Yeah, so question for actually both Dr. Kumar and Dr. Davidenko. When referring to the source model as opposed to the sound model, would Dr. Davidenko's sound swapping, his experiments there, would that align with the source model for explaining misophonia?
I would say fairly sure. I think there also, if I understand this correctly, is kind of the emphasizing of the source more compared to the sound, because the same sound with a different swapping, for example, you can perceive the same sound differently. So the context kind of really matters. I think that's what I feel the crux of the argument there is. So I think probably it is in line with the source argument. Yeah.
Just another question, since we're talking about the people that we want to study. Who do you think is missing from studies of misophonia? And do you have any suggestions on recruitments of people to study to get a more representative sample or diverse sample? That could include obviously demographics, but also maybe comorbid conditions. Do you have any thoughts on that? I guess anyone can jump in on that. I see a couple people nodding, Dr. Hansen and Dr. Davidenko, but
I mean, I can, because we have been. Sometimes definitely we would like to have a more diverse sample, but then again, I think because at the end of the day, we are kind of like doing a group comparison, and then if there's a group, there's a lot of variability, then you don't get the answer. So I think it'll be good to get some sort of like a large sample individualized where we have kind of reaction which varies across the population, and then you do some sort of correlation analysis. We tried to do something similar, for example, in our 2017 study where we took individual sounds reaction. Obviously, it was again categorized into a few categories, but we tried to do an individual subject. But again, I think that wasn't maybe more diverse, you can think of that they were like the triggers were basically based on particular orofacial triggers. They were not other triggers. Now, whether if you want to induce the subject where the type of trigger also varies quite a lot, and then whether there are some individualized areas that they pop up based on the type of trigger they have, you need obviously a very large sample. And sometimes even, for example, in Iowa, actually, we have been, and probably Chris, we have been struggling to get participants actually, because within a local population. And so I think it would be good to recruit and maybe reach to more advertisers to get a more diverse population. But that needs a very, very large sample, and obviously you need lots of funds to do that. Yeah.
Yeah, that's a good question. In terms of just the numbers of people, you're in Iowa. There's research happening in small pockets everywhere.
Yeah.
Have you thought about how to? I mean, Dr. Hansen is going to Montreal; it's a big city, but like how? And I know I've talked to Dr. Storch where they had to obviously fly a lot of people in, and that was kind of tricky during COVID. Have you? Can virtual be an option for certain types of studies? Like, how are you approaching that problem? Anybody can get. Sarah, do you have any thoughts on that?
Yeah, I mean, I think virtual can definitely be an option depending on the type of study. The results I presented today were all done online, so it's definitely an option. Part of the issue with that is that you lose some of the, I think, like rigorous clinical characterization that you're able to do in person, and so you kind of have to weigh those two things. But
if I just mention something related to this kind of diverse sample idea, I think it would also be really interesting to do kind of a cross-cultural comparisons. And there have been studies recently published kind of looking at prevalence rates in different places. And there's, cultural differences that may make one sound a trigger in one culture and not a trigger in another culture. We see that already, even in our relatively small samples. So it would be really interesting to know whether that results in different prevalence rates in different locations, or if the nature of trigger sounds themselves are different in different geographic locations.
Gotcha.
Kind of at time. I don't know if you want to wrap up. I know there's a lot of good questions coming in, but we have a quick Happy Trails event right after this. And
okay. Yeah.
So everybody can go to their hard-earned dinner break in about 10 minutes.
Yeah, that sounds good. Unless there's, I didn't realize we're already at time. Time flies. Well, I want to at least say thanks for everyone for participating. The afternoon session was just as amazing as the morning one. Thanks for all the work you're doing, and we're all excited to kind of meet up again maybe next year or beyond to find more breakthroughs. Thanks everyone coming in. Thanks for everyone watching.
Thanks everyone.
Thank you so much. Thank you.